That just means you are up to watch one of the advanced instruments in biotech research. So what is it? Yes, the instrument that we chose is NMR spectroscopy. NMR spectroscopy is probably the most powerful tool available for characterizing the structures of organic and biochemical compounds. In biotechnology research, NMR spectroscopy serves as a versatile and indispensable tool with several specific functions. One primary application is the structural elucidation of biomolecules. For proteins, NMR provides valuable data on their folded conformations. It helps to understand their active sites, binding interfaces, and overall tertiary structures. This is crucial for unraveling the mechanisms underlying biological functions, as well as for designing targeted modifications or drugs. Moreover, NMR is extensively utilized in the study of nucleic acids, offering insight into their secondary and tertiary structures. Understanding the special arrangements of DNA and RNA is vital for deciphering processes like replication, transcription, and translation. In the realm of carbohydrates, NMR aids in characterizing complex structures such as those found in glycoproteins, contributing to our understanding of cellular recognition and signaling. Biomolecular dynamics is another key aspect where NMR excels. It can reveal information about the flexibility and motion of biological macromolecules. This dynamic perspective is essential for comprehending how proteins and nucleic acids undergo conformational changes during various cellular processes. In drug discovery and development, NMR plays a pivotal role in elucidating the interactions between the potential drug compounds and their target biomolecules. This information guides the design of optimization of drugs by providing insights into binding affinities, molecular recognition, and structural changes induced upon binding. Lastly, NMR spectroscopy is integral to metabolomics studies in biotechnology. It allows researchers to identify and quantify metabolites, providing a comprehensive view of cellular metabolism. This information is crucial for optimizing bioprocesses, understanding cellular responses to environmental changes, and the enhancing of production of bio-based products. Many nuclei has been, and all nuclei are electrically charged according to the NMR principle. An energy transfer from the base energy to a high energy level is achievable when an external magnetic field is supplied. All nuclei are electrically charged and have many spins. Transfer of energy is possible from base energy to higher energy levels when an external magnetic field is applied. The transfer of energy occurs at a wavelength that coincides with the radio frequency. Also, energy is emitted at the same frequency when the spin comes back to its base level. Therefore, by measuring the signal which matches the transfer, the processing of the NMR spectrum for the concerned nucleus is yield.
Hi everyone, so let's move on to the components of NMR. There are three main machines in NMR. For the first one is the NMR magnet, second is the NMR spectrometer console, which includes the radio frequency system, including the transmitter and detector. And lastly is the workstation computer that manages the NMR instrument operation. However, there are a few major components that plays a big role in NMR operation. First is magnet, which generate a homogeneous strong magnetic field that emits electromagnetic radiation between 60 to 100 MHz. Second is magnetic coil that generates a magnetic field when there are a current flows through it. And third is radio frequency system that controls the nuclear spin and detect the signal. FAT is radio frequency transmitter. It produces radio frequency pulses at the predetermined power, frequency, form, and timing. The frequency generator and waveform generator creates the necessary shape. It also features a power amplifier that steps up or regulates the radio frequency power to the predetermined value. In addition to a gate that regulates the radio frequency transmission at a specific time. Next is radio frequency detector. It determines the unabsorbed radio frequency, analog the NMR signal, and convert it into digital format by analog to digital converters ADC. Next, computer. It controls the radio frequency, accumulate, process, and display the data. Lastly, sample holder or sample probe. It holds the sample in the magnetic field and it is equipped with coils for transmitting and receiving the radio frequency signal. So this is the example of NMR magnets that are available in market that calls the SN line. So this SN line have 3 different NMR magnets which may able to achieve different range of frequency. So for the first one is the SN 400 MHz, second is SN 600 MHz, and third is the most powerful, which is the SN 1.2 GHz. So let's talk about the SN 1.2 GHz. It's produced by a company called Bruker and it costs about $17.8 million. Bruker is the world's largest manufacturers that specialize in the design and production of NMR, with SN lines combine several of Bruker proprietary innovative magnetic technologies for best performance and cost effective. It uses advanced superconductor technology, so this NMR able to produce magnetic field as high as 1.2 GHz and minimize the physical size and weight of the design to make it more compact. Being a good screen against external magnetic field disturbance, it can even be used in a challenging location.
The first feature is nuclear magnetic resonance is non-destructive analysis. Non-destructive analysis is NMR spectroscopy is characterized by its ability to extract information from a sample without altering or damaging it. This prevention of sample integrity is particularly beneficial for delicate or limited samples, ensuring accurate molecular level representation. The non-evasion nature of NMR allows for repeated measurements on the same sample, facilitating extensive data collection, result verification, and experimentation under varied conditions without depleting the original material. In research, especially in biology, NMR's gentle approach enables the prolonged study of a single sample, ideal for experiments requiring numerous measurements over time. NMRs ensure the purity and accuracy of sample representation in complex mixture, crucial for maintaining fidelity in results. Furthermore, its compatibility with sensitive samples, such as those in biology or environmental studies, positions NMR as a valuable tool that investigates without causing any harm, ensuring the safety and integrity of the sample throughout the analytical process. Next special feature of nuclear magnetic resonance used as quantitative analysis. Quantitative analysis in nuclear magnetic resonance spectroscopy leverage the direct relation between signal intensity and the number of contributing nuclei in a sample. Each distinct nucleus generates a specific signal, with greater number intensifying the corresponding signal. Integration of NMR peaks measuring signal areas enables researchers to assess the relative abundance of components, providing semi-quantitative or quantitative results. Comparing integrated signal facilitates determining the relative concentration of molecular species, with calibration adding conversion to absolute concentrations. NMR's quantitative nature extends to determining molar ratios, aiding in understanding stoichiometry and characterizing complex. Widely employed in chemistry and biochemistry, it assesses concentrations, reaction kinetics, and metabolite quantification. Combining quantification with NMR's dynamic studies allow monitoring concentration changes over time, offering insights into reaction rates. Despite its quantitative capabilities, careful consideration of factors like retention time and instrumental parameters is crucial for accurate results. Moreover, nuclear magnetic resonance spectroscopy is wide applicability. Nuclear magnetic resonance spectroscopy is a versatile tool with application across diverse scientific domains. In organic chemistry, NMR is crucial for determining the structures of small organic molecules, providing insights into molecular connectivity, stoichiometry, and functional groups. Beyond organic compounds, NMR spectroscopy is equally valuable in studying inorganic substances, which is offering key insights into pollution complexes, metal complexes, and electronic structures. In the realm of biology, NMR serves as a powerful tool for unraveling the three-dimensional structures, dynamics, and interaction of large biological macromolecules such as proteins, nucleic acids, and carbohydrates. Its widespread use extends to structural biology, where NMR is employed to determine the structures of protein and solution, which can aid in understanding their functions, folding pathways, and interaction with ligands. NMR also plays a vital role in nucleic acid analysis, metabolomics, material science, medicinal chemistry, environmental analysis, and quality control in industries such as pharmaceuticals and food, which is to ensure the integrity and purity of chemical products across various applications. The next special feature is isotopic sensitivity. Isotopic sensitivity in nuclear magnetic resonance spectroscopy is pivotal for discerning distinct isotopes within molecules. NMR active nuclei such as hydrogen, carbon-13, and nitrogen-15 with non-zero nuclear spins facilitate this discrimination. Sensitivity to isotopic variations arises from difference in the number of neutrons influencing the nucleus magnetic properties. Notably, hydrogen isotopes such as protium, deuterium, and tritium are exhibit unique NMR signals due to varied magnetic moments. NMR enables quantitative isotope analysis by comparing the signal intensities, offering insights into isotope distribution within molecules. Techniques like nuclear Oberhauser effect spectroscopy leverage isotopic sensitivity for spatial proximity information between nuclei. Stable isotopes like 13 carbon and 15 nitrogen, commonly used in NMR, facilitate selective observation through labeling, providing specific details about molecular regions. In biochemistry, isotopic sensitivity aids in studying biomolecules, while in geological and environmental studies, NMR sensitivity to isotopic composition informs about geological formation and environmental contaminant sources. Another special features of nuclear magnetic resonance spectroscopy is high resolution. Modern NMR instruments provide high spectral resolution, crucial for analyzing complex mixtures. This capability ensures well-separated peaks in the spectrum, facilitating the identification and quantification of individual components, particularly valuable in fields like metabolomics and environmental analysis. High-resolution NMR is indispensable for studying subtle structural features, which is observing small chemical shifts and understanding coupling patterns, offering detailed insights into the chemical environment of nuclei. It is especially beneficial for analyzing isotopic fine structure, which can detecting subtle shift due to isotopic variations, contributing to a more comprehensive understanding of molecular compositions. The wide dispersion of chemical shift in high-resolution spectra is vital in chemistry, which can allowing the differentiation between various functional groups within a sample. The last special features of nuclear magnetic resonance spectroscopy is versatility. Nuclear magnetic resonance spectroscopy's versatility spans diverse scientific domains. In chemistry, it elucidates molecular structures, while in biochemistry, it probes biomolecular intricacies. Its application extends to medicine for non-invasive imaging and material science for molecular-level material analysis. NMR transcends compound limitations, embracing small organics, inorganics, polymer, proteins, and nucleic acids, marking it as a universal research tool. Its dual capacity for quantitative concentration determination and qualitative structure identification fosters comprehensive analysis. The non-destructive nature of NMR permits repeated measurements, which is very suitable for varied experimental setups. Customizable experiments employing different pulse sequences cater to specific research goals. Ongoing innovation in NMR instrumentation, including hardware and software advancements, continually broaden its capabilities and applications. Nuclear magnetic resonance spectroscopy is a versatile and powerful analytical technique with a wide range of applications. In the field of chemistry, it is used to determine molecular identity and structure, which is able to providing insights into the physical, chemical, and biological properties of matter. NMR forms the basis for magnetic resonance imaging, which is MRI, which is a non-invasive diagnostic tool used in medical imaging. In drug discovery, NMR spectroscopy plays a crucial role, facilitating trials of cancer therapies, optimizing nutritional supplements, and providing information of molecular interactions. It is also a powerful technique for the characterization of small molecules and determination of their structures. In the fields of biochemistry and biophysics, NMR spectroscopy is used to unravel protein structures, analyze amino acid profiles, and study molecular mobility, which are contributing significantly to advancement in understanding biological processes. Additionally, NMR spectroscopy is employed in hit finding in disease states by targeting protein structures and aiding in the identification of potential therapeutic targets, which is offering promise in treatment development.
NMR spectroscopy is a crucial tool for characterizing organic and biochemical compounds. It provides data on folded conformation, peptide, binding interface, and tertiary structures of biomolecules. NMR plays a key role in drug discovery and development by elucidating interactions between potential drug compounds and their target biomolecules. It's integral to metabolomic studies, allowing researchers to identify and quantify metabolites. It also a non-destructive method extracting information from a sample without damaging it. NMR also useful for analyzing bond, connectivity, stereochemistry, and molecular conformation. Quantitative analysis uses the correlation between signal intensity and nuclear abundance. NMR also captures spin lattice and spin-spin interactions, detects chemical exchange, and probes molecular dynamics on various timescales. NMR widely used in organic chemistry, organic compound structures, and quantitative analysis. It also offers insight into molecular flexibility and molar ratios.